Thank you for joining me once again for AJP Heart and Circ from the editor's desk, video highlights. This week we have some great science for you to enjoy, so let's get started. First, in a review associated with our call for papers on mitochondria in cardiovascular physiology and disease, Hollander, Thapper, and Shepard summarized the evidence for inherent structural and functional differences that exist between spatially distinct cardiac mitochondrial subpopulations. This distribution of mitochondria close to various intracellular organelles may have implications for understanding cardiac pathology and intracellular energy metabolism. In a study targeted to understand the role of the local renin-angiotensin system in the response of human microvessels to pressure, Durand and colleagues show that treatment of these vessels with either angiotensin receptor antagonists, an ACE inhibitor, or the antioxidant enzyme SOD prevented endothelial dysfunction in response to pressure overload. This study has implications for vascular damage following multiple exposures to high pressure. Calcium-sensitive dyes have been used for many years to evaluate myocardial function. In a potentially important study, Kong and Fast compared calcium optical signals using dyes with varying affinity. They concluded that calcium transients in ventricular muscle are strongly correlated with dye affinity. Finally, Buchholz et al. Provide data showing the decrease in dystrophin levels during myocardial ischemia is mediated by an increase in MMP2 activity. Using isolated rabbit hearts, these Argentinian investigators concluded that dystrophin is a novel substrate for MMP2 and that ischemic preconditioning may prevent dystrophin breakdown by inhibition of MMP2 activity. I want to remind you that this is just a smattering of the great work published this week in AJP Heart and Circ. Go to the table of contents for more great science. See you in 2 weeks, and thanks for listening.